This is my first trip to the Simons Foundation, so I'm very excited to be here and to talk with many of you afterwards. And I love the old school pointer that they provided me with, so I'm going to use it. My earliest work began, as Alyssa mentioned, in these episodic disorders, and we also did a lot of work in hereditary neurodegeneration. This work began when I was a neurology resident at the University of Utah, and I worked in the laboratory of Ray White, using the emerging tools of modern human genetics to map and clone human disease genes. What I'm going to talk about today is circadian rhythm and sleep traits. These are behavioral traits, and this is behavioral genetics. And it's decidedly a paradigm shift from moving from disease genetics to behavioral genetics. Up until this point, I would argue that behavioral genetics in humans had been completely unsuccessful. And for that reason, I stayed away from it until a point at which I'll tell you about in a moment. So genes and behavior is really, really hard. And why is that? Behavior is really complex. I'm a geneticist, and I believe that everything is genetic, but it's also true that everything is environmental, and it's the interaction of what we got from our parents, plus what we experience in the uterus, in development, when going to school, in our families, in our societies, that modify the expression of the blueprint that we got from our parents. In the sleep system, circadian and sleep homeostatic systems, we really began to muck around with our biology with the invention of electric lights, when we could have bright light late at night or early in the morning before the sun had come up or after the sun had gone down. We use alarm clocks. We have all kinds of family, school, and work responsibilities. We ingest compounds that affect our sleep, like coffee and alcohol, and some of the medications that we take. Our emotional state and our feeding behavior affects our sleep. And so teasing out what's genetic and biological about our sleep is a real challenge with human sleep behavior and with other animal sleep behavior compared to trying to decide whether someone has Huntington's disease or Alzheimer's disease, for example, which as a neurologist I can tell you is a lot easier than phenotyping for behavior. So it's not surprising, I would argue, that all elements of our physiology, they certainly have all co-evolved with our life and our evolution on this planet with a 24-hour circadian day. And so physiologies that include neurodevelopment, affective behavior, sensory systems, immune systems, wakefulness and consciousness, and even cell growth, cell cycle regulation, these are all tuned and have evolved to work optimally in an environment that's changing on a 24-hour, in a circadian way on a 24-hour basis. Consequently, desynchrony from the solar day or chronic sleep deprivation leads to increased risks, I would argue, of virtually all diseases. Certainly neurodevelopmental disorders, depression, pain, migraine headache, autoimmune disorders, neurodegeneration, and all cancers that have been looked at. So sleep is really, really important. But it remains true that we don't understand at all what sleep is. We know a little bit about synaptic pruning and memory consolidation, but the biology of normal human sleep is really still very much a black box. And what we'd like to do is change that. I'm going to talk about primarily two phenotypes today. I'm going to start with our work in advanced sleep phase. We've known for a long time, you've known for a long time, that there are people who are morning larks and people who are night owls. And so that this is not a novel thing. But what was our entree into this field, and the reason that I decided it was time to get into behavioral genetics in humans was when this 69-year-old woman presented to my colleague, Chris Jones, who's a sleep neurologist at the University of Utah, and she complained that she didn't like waking up when it was cold, dark, and lonely. So she's been waking up. I talked with her a couple of years ago, and she's well in her 80s now. And she's waking up at 2 o'clock in the morning, and she's so tired that she will fall asleep at the dinner table. And so people told her she was depressed and that she was antisocial. But the fact of the matter is she has very strong ASP, advanced sleep phase. And as Chris Jones and I set out to characterize this family, we recognized that this was an autosomal dominant trait in her family. And this made me excited that maybe we could apply modern human genetic tools to map and clone human disease, or to map and clone human circadian genes. You can see that about 50% of the children of affected individuals are affected. The filled symbols, the shaded symbols are affected individuals. The ones with dots in them are people who are unknown. They're too young to be certain, or their phenotype might be sort of morning lark, but not strong enough to call them definitely affected. All during this time, we had to define the diagnostic criteria for this trait that we were setting out to study, because it's critically important that we know who's affected and who's not and who's unknown if we're going to try to understand the genetics of a trait. Well, I just want to make the point that in the general population, there's a huge variation in what all of your biological tendency to sleep is. And how you really sleep may be quite different than your biological tendency. But if we strip away alarm clocks, electric lights, coffee, alcohol, stress at work, school responsibilities, etc., and we say, okay, some of you are, programmed to wake up at 7, and some are programmed to wake up at 9, based purely on biology. Most of the variation in the normal distribution here it's still genetic, but it's complex genetics, and it's probably the summation of many variants of small effect in many genes that lead some of you to be a moderate morning lark and some of you to be more of a night owl and want to sleep later. So FASP, advanced sleep phase, is ASP. Those are the morning larks. Their sleep phase is advanced relative to the solar day. And so as geneticists, and this is true for people like Justin, who works in Drosophila, and people who work in any organism, we like to look at the extreme phenotypes, because when you look at extreme phenotypes, they're more likely to be strongly genetic. Well, that family that I showed you is much bigger. We've expanded it from the pedigree that I showed you. And so we have lots and lots of people. We've cloned the gene, we know the mutation. And if we look at all the people in that family who have the mutation causing familial ASP, FASP, the distribution of wake-up times in those affected individuals is quite varied. Well, we were able to be successful in the genetics because the mean of this normal distribution of these subjects who have mutations is shifted so far to the left from the general population that we had the power to go after and identify the gene. But the variation that occurs among all the mutant gene carriers within a single FASP family, that's probably reflecting the genetic background that's causing the variation in the middle of the distribution for the general population. Does that make sense? Okay, so I'm going to talk about circadian clock. This is the timing of sleep. And then at the end, I'll come to talk a little bit about sleep homeostasis. And sleep homeostasis is about how much sleep you need and how sleepy you are as a function of how long you've been awake. So if you're awake for 24 hours, even if your circadian clock is telling you to get up, that it's time for your day to begin, you can still override it and fall asleep because you're sleep deprived. And these two processes are interacting in us to reinforce healthy sleep patterns. And I'm not going to talk about details, but we've spent many years characterizing in great detail biochemical regulation of the circadian clock. And the first gene which we cloned, which is PER2, is the substrate for a kinase, which was the second gene we cloned, casein kinase 1. And the mutation in that large Utah family that I showed you occurs at residue 662. There's a serine there that's mutated to a glycine in this family. And what we showed back in 2007 is that CK1 is having opposite effects through modifying the substrate PER2 in different regions. So the same enzyme is working at different places in this protein that have opposite effects from one another, which seemed sort of wasteful to me at first. But I've come to believe that this is part of nature's way to deal with homeostasis, to how to balance the length of the period is all critically dependent on whether you tip the scale. Oh my gosh, it has a mind of its own. If you tip the scale toward more phosphorylation at this site versus more phosphorylation at another site. And so, and then we went on to show that O-gluconate transferase and O-gluconase, two enzymes encoded by two genes, are novel circadian genes that hadn't been recognized in any other model system. And these enzymes are important for glycosylation, O-linked glycosylation of some serines and threonines. And Chris Ptacek in Ying-Hui's lab showed that the very same serines that are phosphorylated by CK1 can also, that there's competition for occupation of those residues with gluconac, and so this is a separate enzyme system that's competing with the phosphorylation system, casein kinase 1, for the same residues. And this has led to work that I'm not going to talk about other than to say it has real implications for diabetes, which is one disease I care a lot about. And so diabetes and poor glucose control changes the speed of the clock. And we also know that abnormal circadian function contributes to increased risk of diabetes. So this is a very direct link of the circadian clock to metabolism. And we know that metabolism and circadian clock are very, very importantly interacting in important ways. I'm going to skip that because it didn't show up right. So what we can do when we clone human genes and identify mutations is put them into flies or mice and see if we can recapitulate the phenotype. So here's the kind of experiment we can do with mouse rest activity behavior. This is a plot of 48 hours. It's a double raster plot over 3 weeks of time where the lights are going off and on and off and on every 12 hours, sorry. So this is one day and then a second day. And this keeps repeating, going down through 3 weeks of time. So the slope, the activity is shown in the black ticks here. This is being recorded by a computer, the wheel revolutions of the mouse running on a wheel in the cage. And this is a vertical line, which means that the mouse is living on a 24-hour day when the lights are going on and off. When we turn the lights off, dark, then the mouse starts drifting a little each day and getting up a little earlier and getting off the wheel a little earlier than the day before. And that's because a normal wild-type mice of the sort that we use have a circadian period that's just less than 24 hours. And they have to adjust a little bit, delay it by 0.3 hours to live on a 24-hour day. Humans are just the opposite. We tend to have, on average, a period of about 24.2 hours, and we have to advance a little each morning with bright sunlight, hopefully, to live on our planet's 24-hour day. This is why I think it's harder for most humans to wake up earlier than it is for us to stay up later, because our clocks are a little bit longer than 24, or the period of our clocks is a little longer than 24 hours. Well, when we took mice and then we took the human gene for PER2, which is mutant in that first family, and we put two or three mutant copies of the human PER2 gene into mice that have their endogenous mouse PER2 genes, the human mutation in the human gene has a dominant effect in causing period shortening in a mouse background that still has normal mouse PER2 genes. And so I should mention that we did temporal isolation in one subject from that family, the proband, the 69-year-old woman, and she had a period that was significantly shorter than and less than 24 hours, and much shorter than the average in the human population. Now, we also speculated that because of phosphate, a modification of this serine to have an inorganic phosphate, which introduces a negative charge, that if we mutated the serine to a negatively charged amino acid like aspartate, that it would have the opposite effect, and indeed it does. So these mice now have a period that's longer than 24 hours, 24.5 hours. This is a single mouse, but we look at groups of mice and calculate statistics and standard deviations. And then when we cross the mutant human transgenes that are in this mouse and this mouse onto a background from mice that where the mouse PER2 gene has been knocked out, the period is even shorter and the period is even longer for the aspartate mutation. So this protein is about 1,000 amino acids long, but the charge on this one amino acid is critically important. And if it goes from -1 to 0, that can affect the length of the period by more than 4 hours. So that, I found that to be incredibly remarkable. And so the balance of the CK1 activity in phosphorylating CK1 is really critically important to humans and other mammals to live on a 24-hour day. And so shown here are populations of mice in this plot, 10 and 10, mutant mice in red, and they're getting up when the lights are still on. No self-respecting C57 black 6 mouse would do that. The control mice wake up and get on the wheels when the lights go off. They're nocturnal, remember. They run all night, or much of the night, and then get off the wheels as the lights are coming on. The mutant mice, like the humans, are phase advanced. Their activity rhythm, and we can do EEG and prove that this is really sleep and wakefulness. You know, a mouse that's not on a wheel could be awake but inactive. But in fact, these mice are waking earlier, running on the wheel, and getting off the wheel and going to sleep earlier than control mice. So this beautifully recapitulates the phenotype that we see in the human subjects. And then, because we've now been studying this for, almost 30 years, we have a huge collection of families and a lot of data, and we have a population prevalence estimate for FASP, these extremely strong morning larks where the trait travels in families from generation to generation, of 0.3%. So nearly 1 in 300 people have this. And it was only first reported in 1999. Why was that? Because most of these people don't have a problem. The 69-year-old woman really didn't like waking up when it was cold, dark, and lonely. But one of her brothers is a very successful businessman, and in Utah was, trading stocks at, 5 in the morning or 6 in the morning or something, and making loads of money. And so he likes this trait. So this again speaks to the fact that this is behavior. It's not a disease like ALS. We won't argue about whether or not ALS is a problem. It's bad to have ALS. But to be a morning lark, some people like it, some people don't like it, and many of the people in the family just say, oh, that's just how I am. So it's much more common than we had appreciated, and consequently, we've been successful in collecting many, many families, now probably approaching 200 families. Well, what are the challenges of studying circadian and sleep in humans? Well, for one thing, mutations in genes with detrimental effects, with severely detrimental effects, have been selected against. So we're not going to be able to find the really bad mutations that we could make in mice or flies in the laboratory. So there's a ceiling effect in terms of how big a change is tolerated, because these people are living and surviving on this planet. And it's also worth noting that everything we do in humans is incredibly challenging, labor and resource intensive, because, as I said, phenotyping behavior is much harder than deciding whether someone has Huntington's disease or not. Well, there are also opportunities, and this is the same as the one on the previous slide. Mutations in genes with detrimental effects have been subject to selection. So we're finding mutations in the circadian clock and in the sleep homeostat that don't cause serious diseases in these people. And we also have a huge amount of additional data that you can't get from flies or mice, in that humans can talk to us. And they have medical records that we can access and learn about their affect and about their anxiety and about what medications they're on and lots of other things. So I will say at this point that most people with FASP, as far as we can tell, are not at increased risk for other problems. And this, again, is consistent with the fact that they've survived on our planet through evolution. And so if FASP also always caused childhood cancer, if FASP genes also caused childhood cancer, it would be far less common than this, because people would die and not be able to pass the genes on to others. This is the second, but now I'm going to tell you about some exceptions to that point I just made. This is the second gene that Ying-Hui's lab published in 2005, the casein kinase 1 delta. And years later, we had collected additional families. The mutation in that first family is a threonine to an alanine in this highly conserved region, the catalytic domain of casein kinase 1 delta. But now we have collected 2 additional families that had mutations in one family 2 amino acids downstream that led to a H to R mutation and a methionine to leucine mutation in a third family at this position. And what was interesting is that all 3 of these families have FASP, but all the mutation carriers also have migraine with aura. And so having 3 different mutations at highly conserved residues in the same gene in 3 families who all have FASP plus migraine with aura argues very strongly that migraine with aura is caused by mutations in this gene. And we've gone on to show that other physiologies that we can measure in the mice that carry those mutations have things like decreased thresholds for inducing cortical spreading depression, which the details of which are not important, but which is a physiology that everybody believes is critically important to migraine pathogenesis. So here's a case where circadian gene mutations in this gene lead to FASP, but they also cause a problem. migraine headache, which again, we wouldn't have been able to, we wouldn't have had a clue to look for in mice if we had initially identified this mutation in a lower organism. Another interesting finding was by Luoying Zhang, a postdoc working with us, that a mutation in period 3 that causes FASP in a human family also, the affected individuals in this family also have seasonal affective disorder. Now, seasonal affective disorder is very common, so this could be purely coincidental. But Luoying proved, first of all, that when she made a mouse model of the human PER3 variant, that those mice had a circadian phenotype, showing that it is, in fact, an FASP gene. In addition, though, we can't really test whether a mouse is depressed, but there are assays for depression-like behavior in mice, and there are different assays, and Luoying applied all of them to the mutant mice and showed that the mutant mice had increased depression-like behavior relative to their non-mutation siblings. So that was pretty interesting, but the thing that really got me excited was that then she put those mice, ordinarily we maintain them in 12 hours of dark, 12 hours of light, a 24-hour cycle. Then she turned the lights off, or no, I'm sorry. Then she changed to a more winter light cycle, like you might have in Alaska, with dark for 20 hours and light for only 4 hours. And when she did that, the mutant PER3 mice had even more significant worsening of the depression-like phenotype. So we now have a paper that's submitted that looks into the biology of this in much greater detail, and we think that this is being mediated through a serotonin signaling system. But another interesting case where a circadian variant, in this case, does have an associated phenotype that we believe is caused by the mutation in period 3. So we think period 3 is sitting at a nexus between sleep and mood regulation. There are 3 genes and proteins in us humans and in other mammals as well. And PER3 is the most unlike the other, is much more unlike the other 2. And it's sort of been blown off by some people as even being a circadian gene at all. But it clearly is a circadian gene, and it clearly has evolved a different function, we think, at a nexus between sleep and mood regulation. So a lot more work to do there, but another case where FASP and seasonal affective disorder seem to both be caused by a mutation in the same gene. So where are we today, or let's say 5 years ago? 5 years ago, we had about 150 families. I told you that we have prevalence estimates of about 0.3%, 1 in 333 people. And we have human families and have identified 6 different genes in which mutations cause FASP. In some cases, we have multiple alleles in the same gene, 3 different families, all with 3 different mutations for both PER2 and CK1, 2 for timeless, and so forth. But what's really interesting about this is that of these families that have all undergone sequencing of all of the candidate circadian genes, less than 15% of them have mutations in known genes, which means that there are either core clock genes in humans that have been missed, or that there's a whole nother category of genes, and I'll tell you what I think that category is in a moment. So beginning 5 years ago, we took our 34 best unexplained families, and we did whole exome sequencing, and we now have 3 novel FASP genes that are mutant in 3 families, 7 families, and 6 families. So again, allelic series in highly conserved genes that really proves genetically that these are causative FASP genes. And I'll tell you in a moment about one of these. Now, most of these, it's not entirely that simple, but most of these have their effects on causing FASP by shortening the circadian period. And again, the details of that are not important other than to say that, yeah, we predict, people long before us predicted that a short circadian period would cause phase advance, a long circadian period would cause phase delay. And that is clearly true, as I showed you in the PER2 mouse data. So if I were to do whole exome sequencing on anybody in this room, and myself, you and me, there would be millions of differences between you and me. And that's true for everybody in this room, and each of you pairwise, because there's this rich genetic variation in the background of normal humans and other organisms as well. So then what do you do with all that data, and how do we figure out what is really the causative FASP gene? And this is the process that we use. We prioritize by saying, okay, FASP is 0.3% of the population. Any one gene isn't going to explain all of FASP, so the frequency of a mutant in the general population has to be well under 0.1%, 0.01% to possibly be an FASP gene. So we weeded out all the variants that occur in the general population at 40% or 30% or 5% or even 1%. We also sequence multiple affected multiple FASP individuals from the same family, from each family. And if it's a causative mutation, then all of the affected people that we sequence should have the same mutation. If not, that would suggest that it's a genetic variant in that person, but has nothing to do with the FASP. And then the most powerful argument, again, is having an allelic series. 34 families, if 7 of them have mutations in the same gene, and it's a gene that is not very tolerant of genetic variation in the general population, that constitutes genetic proof that this is a causative gene and these are causative mutations. And that was true for the 3 novel FASP genes that I showed you, and I'm going to tell you about one of them now. We're preparing a paper to submit on this topic presently. So in the calcium channel CAC1A1D, it's a neuronal voltage-gated calcium channel. So this is completely different than the episodic diseases that I studied when I was younger. But quite remarkably, this gene is homologous to the second human muscle disease gene that I had cloned in 1994. So that was kind of fun. But these are highly conserved genes with 24 that encode proteins with many membrane passing segments. So they don't tolerate mutation very well. And yet we had 6 different mutations in 7 families for this gene. There was one family that had, there were 2 families that had the same mutation and 5 others that had all novel variants. And here's the frequency of those variants, 4 in 100,000. The exact database has hundreds of thousands of whole exomes in it now. And so 4 of these are variants that have never occurred in the general population for those hundreds of thousands of people who have undergone whole exome sequencing. And these are a little bit more common, 4 in 100,000, 2 in 10,000. And this is just to show the families there. In each of these cases, all 3 of these individuals, all affected with a trait, had the same mutation, and the unaffected individuals in the family didn't. And the same is true for all the other families. So novel variants, novel or rare variants, segregating in the families, and they're predicted to cause amino acid changes at these positions scattered throughout the protein. But what's really interesting to people who think about calcium channels a lot is that the C-terminus and this loop between domains 1 and 2 are critically important in C-terminal inactivation. So this is a C-terminal inactivation domain for this calcium channel, and it interacts with this part of the protein on the inside of the cell to close the channel after it's opened. And so we set out to collaborate with Dan Miner and Fayal in his lab. And again, I'm not going to go into the details, but all of these mutants have functional consequences on calcium channel currents when we put them into cells growing in culture and do patch clamp analysis. So they have, you know, it's probably true that some polymorphism, some variations in our, it's certainly true that some of the variations in our genes cause small functional consequences, but don't really cause a phenotype. In this case, all of these variants cause functional changes, and that doesn't prove that they're causative, but as I said, the genetics was quite conclusive in that regard. So we took the one that gave the most severe changes in calcium conductance, and we made, because we didn't want to make 6 different animal models, we made one animal model for this variant, the one that in vitro had the most severe functional consequences. And this is a slide simply to show that the circadian period was not significantly different between the wild type mice and the mutant litter mates. So, and this is something that we had predicted. It may be that there are either primate-specific circadian genes that weren't found in Drosophila and mouse, or more likely, I think, because the assays in flies and mice that have been done are different than what we do to phenotype humans, we thought that it might be the case that mutations that affect entrainment of the circadian clock, not the speed at which it's ticking, but the sensitivity to shifting forward with bright morning light or shifting back with late daytime light, might be different. And so this mouse model showed us that the period was not different between wild type and mutant mice. But remarkably, when we gave a light pulse shown in blue here and then measured the effect on how quickly the mice were in training, quantified here, there was a statistically significant difference. It was stronger in the males than in the female mice, but in all the mice, a statistically significant shortening or a phase advance, changes in entrainment that would be consistent with a phase advance. So the folks who were doing screens in Drosophila weren't looking at phase, they were looking at period, because it's easy to measure rest activity behavior and then determine the period of an animal. We can't do that. Doing temporal isolation in humans is a Herculean undertaking. So we are focusing on phase. And the predictions by circadian biologists prior to all of this work were that you could get phase advance either by period shortening or by altered sensitivity to sunlight in the morning and sunlight at the end of the day to advance the clock or delay the clock. And that, in fact, is true, and we've done formal phase response curves to prove that in this mouse model. So I think FASP is opening a whole new area. A majority of our FASP families don't have mutations in known core clock genes. I think the genes, and we have 2 more identified, and we have a whole lot more sequencing we'd like to do, but I think many of these mutations are going to affect the sensitivity to entrainment, and that this will be a exciting area to mine in the coming years. Okay, now circadian rhythms exist in all of our cells, but the master circadian regulator is a very small part of the human brain and the hypothalamus, 10,000 neurons on each side. And all you have to do is cut this, so go in and do surgery and damage this part of the brain on both sides in a mouse or a rat or a non-human primate, and you can make them completely arrhythmic. So the anatomy of the core circadian oscillator is very simple. We know where it is, and we've known that for decades. Now I'd like to talk about sleep, which is, I think, a much more complicated, much more interesting, but also much more challenging area. So here I'll turn to families that we've been collecting over the last 15 years that have natural short sleep. These are individuals who sleep 4 to 6 hours per night, and they feel great. They don't complain of being sleepy. They feel good, and they in fact say, well, if I stay in bed for an extra hour and try to sleep an extra hour, I feel real crappy. They feel crappier after sleeping 5 hours than if they sleep 4 hours. And that's decidedly different, I think, than most of us humans. How much sleep do we need? This slide will look familiar to you. Everything is genetic. How much sleep? There's some people who say everybody needs 8 to 8.5 hours of sleep. That's as ridiculous as saying that all of you should be 5'10", and those of you who are shorter are diseased, and those of you who are taller are diseased, right? No, that's just not how it works in human genetics and biology. In the general population, we all have slightly different needs for sleep. But again, as geneticists, if we look at the tails of this distribution, there are people who need 11 or 12 hours of sleep. There are plenty of lazy people in the population. But if you weed those guys out and you say, oh, this guy needs 11 or 12 hours of sleep, they work hard and they're motivated and they're type A, but they really feel bad if they don't get 11 hours of sleep. There are people like that. Again, a majority of us fall into a range where we need 7 to 10 hours of sleep. But there are these remarkable elite sleepers, the natural short sleepers, and as well as natural long sleepers, which we're also interested in. But I'm going to just talk briefly about natural short sleep here. And similarly to FASP, I would argue if we had big enough families and if we had more data, that it's probably true that the amount of sleep needed by mutation carriers in the same family is going to vary considerably, but the mean is shifted way over here, 4 to 6 hours. So there will be some mutation carriers who need closer to 6, some who need 4, some who need 6.5. And that's because, I think, of the genetic variation that's conferring this kind of variation in the general population. Well, model systems are being used to identify sleep genes, and I have to warn you now that what I'm going to say will be deeply unpopular with some individuals, but I want to start here. Alzheimer's disease, common neurodegeneration. Many of these patients have sleep disorders, and in fact, that's the #1 reason for institutionalization of Alzheimer's disease patients, that the sundowning that the caregivers just can't deal with. They themselves are horribly sleep deprived. Most patients with Parkinson's disease have disrupted sleep, and it's different than the Alzheimer's disease patients. And the most common complaint of ASD families, autism spectrum disorder families, is the disrupted sleep of the children with ASD. This is very, very problematic for the families in terms of caring for these children. So then I ask this question: are these all sleep genes? These are familial Alzheimer's genes, presenilin 1 and 2, ApoE4, APP. These are Parkinson's disease genes, PARC2, 7, alpha-synuclein, LRRK2. And these are genes that have been implicated in autism, Fragile X, SHANK3, SYNGAP1, and others. Are these sleep genes? No, they're not. They're genes that cause disorders of the brain. And the problem in studying sleep, I think, in the genetics of sleep, is that the anatomy of sleep is far more complicated than the anatomy of the circadian system. These are all nuclei in the deep brain that are known to be important in different aspects of wake and sleep behavior. And these are additional areas, including regions of the cortex that are important for sleep. So sleep is a trait whose biology and circuitry is distributed across large parts of the brain. And so it's not surprising that brain trauma of many sorts, that strokes of many sorts, that Alzheimer's disease and other neurodegenerations, that neurodevelopmental disorders all have sleep problems, because you only have to mess with some part of the brain that's important in the sleep network in order to get a sleep phenotype. And this, I would argue, is why the first sleep gene in Drosophila, which is the shaker gene, cloned by my friend and colleague Lily Jan 40 years ago at the University of California. This was called a sleep gene, but these flies are hyperactive. They have epilepsy-like episodes. They die at a very young age. And so are potassium channels important in sleep? Absolutely, I believe they are. But is shaker a sleep gene? I'll see what Dr. Blau thinks about that. But I would argue that it isn't, that it's a brain disease gene. So that's why I'm so excited about these natural short sleepers, that they are going to really be what helps us to understand what normal human sleep is all about. And why do I think that? Let me just pull these all up quickly. Individuals with natural short sleep not only don't have any recognized additional problems like neurodegeneration or bad behavior or difficulties with social interaction, they appear to be healthy. Many of our subjects are in their 80s and 90s and are really quite healthy. We've shown in studies of our families that people with FNSS have higher resilience to all kinds of different traumas, emotional and physical. They are, and every clinician who has worked with these subjects and every clinical coordinator over the years that's worked with these subjects says they're incredibly optimistic, they're positive, they can do it, they figure out how to do it. Often they have two jobs, not because they need the money, but because they want to keep busy. They have high behavioral drive, and they suffer effects of jet lag much, much less than average. And so I think FNSS is a very positive, or it has many other positive behavioral attributes to argue that it is not a disease gene. It's not a brain disease gene disrupting sleep. It's a gene and a mutation in a particular family that allows those individuals to sleep a lot less and to do really, really well. So the first 4 genes that we've cloned and published are shown here. The first of them, which Ying-Hui published in 2010, is DEC2, a transcription factor. This, in fact, is a circadian gene also, I should say. And so we think DEC2 has co-evolved in both the sleep homeostat and the circadian clock to affect the regulation of both of these different processes. The subsequent 3 published genes are all GPCR, all encode GPCRs, adenosine receptor B1, the neuropeptide S receptor, and the metabotropic glutamatergic receptor 1. And these are all druggable targets, so that's pretty exciting. Now we have 3 more FNS genes that have been identified. We're struggling to get funding to study the mouse models that we've generated. And the really exciting thing is that from our 120, 140 FNSS families, that these 7 genes, 3 published and 3 more as yet unpublished, explain only a small minority. And the reason I think this is exciting is that understanding sleep, I would argue, is like a 1,000-piece jigsaw puzzle. And you have the box of the jigsaw puzzle and it doesn't have a picture on the front. That's a problem. So one of the things that we're doing is we're trying to clone and identify as many of these genes and as many mutations as possible. And then as we get pieces of the puzzle, this one with red flowers, and this one with red flowers, and this with a checkered tablecloth, or whatever, that a picture will begin to emerge, and at the same time, we're interested in looking at where all of these, where expression of these genes overlap, because those would be parts of the brain that we predict are important for sleep, and biological pathways, where do these different receptors, and in this case transcription factor, where do they converge in terms of their biological function? Because again, that's going to give us important clues about what normal human sleep is, where its function is subserved in the brain. And all of this work has been done. Chris Jones recently retired from the University of Utah, but he's a sleep neurologist and colleague when I was at the University of Utah. Ying-Hui Fu, who's at UCSF, who's been an active collaborator, and she really has driven all the natural short sleep work. But this is why I think it's really important. You know, if we're trying, I have never worked on Alzheimer's disease, nor have I done studies to try to develop treatments for Alzheimer's disease. But this is a really important area as we get older. As I get older, I care more and more about this. But the problem is, once you have Alzheimer's disease, the horse is out of the barn. And fixing the problem after a lot of neurodegeneration has happened is going to be really a hard problem. We think that if we can intervene in helping to make people more healthy by improving their sleep, that this might be a more effective way at delaying the onset and the rate of progression of diseases like Alzheimer's disease. So I've told you, natural short sleepers, these are some slides from Ying-Hui, sleep 4 to 6 hours a day for their entire life. They're active and healthy in general. We have these mutations. And so Ying-Hui had hypothesized early on that maybe the natural short sleepers, because they look so good, they do so well, maybe they're just doing everything you need to do when you're sleeping more efficiently. You know, what does that mean biologically? We don't know. So we know that poor sleep leads to more rapid progression of Alzheimer's disease. We know that neurodegeneration leads to worsening of sleep. So this is a vicious cycle, bidirectional process where poor sleep contributes to this contributes to poor sleep. And so some of our colleagues predicted that if we were to cross Alzheimer mutations onto a natural short sleep genetic background, that the mice would get sick earlier and faster and progress more rapidly. And so Ying-Hui had a postdoc in the lab who set out to test this. Do the FNSS mutations offer protection from disease? So she crossed, first of all, the DEC2 mouse, FNSS mouse, against two different models of Alzheimer. There are hundreds of models, or over 100 models of Alzheimer's disease in mice. And two very commonly used ones are the A-beta 5XFAD or the PS19 tau mouse. And so what was really gratifying to Ying-Hui, having made that prediction, is that here you see in the PS19 mice, green shows tau accumulation. That's a bad thing if you have Alzheimer's disease. And then when they cross the PS19 mutation onto the DEC2 mutant background, there was far less tau accumulation, and that's quantified here. And the same was true when DEC2 was used for the 5XFAD model. There was much more A-beta accumulation than in the DEC2 mutation onto which this Alzheimer's disease gene was crossed. And so that's just one FNSS model, but we have 7 now for which we have mouse phenotypes. And so the next one that Dongqing set out to study was NPSR1. When she crossed the PS19 mutation onto the NPSR1 mouse, the PS19 alone causes lots of tau accumulation, and the heterozygous NPSR1 FNSS genetic background leads to a dramatic decrease in the accumulation of tau, quantified on the right. And the same was true for the 5XA FAD mutation when crossed onto MPSR1. And the same is true, I won't show any data, for the GRM1 mutation. So this really argues that natural short sleepers are doing something fundamentally different. And what is the importance of sleep for brain health? Well, we talk about, a lot of people talk about clearance of toxins, energy homeostasis. These things all make sense, but we have to study them, and other peoples are studying them in Alzheimer's disease. But if indeed it's the case, then we can hope to understand what it is about sleep and sleep efficiency that leads to these remarkable elite sleepers and the effects that these natural short sleep variants have on prolonging the onset, delaying the onset of Alzheimer's disease pathology. These are all the folks I've tried to mention along the way who have contributed to this work and funding from the Bose Foundation and NIH. And I'll stop there and be happy to take any questions. Yes, sir.
Oh, so two questions. You talk about the circadian rhythm of 24 hours, but there are humans and animals that live in northern latitudes where that's not the case. So is there any...
Yeah, so animals living at northern climates are still living on a 24-hour circadian cycle, but your point is well taken. The day length and night length is quite different at the North Pole, at the South Pole, and everywhere in between. So we don't know, I've not done any work, I don't know the literature on circadian studies in caribou. I'm sure people have looked at this. But it's not that they don't have circadian rhythms, it's just that they have had to adjust in different ways to more dramatic changes in the length of dark and the length of day.
Like Eskimos, for example?
Presumably, Eskimos, indigenous people of Alaska, let's say, I wouldn't be surprised. We don't have any data. We don't have any Inuits in our collection of FASP families. But presumably they have different mechanisms, or they have, I have a postdoc who's working on metabolism and is looking at changes, seasonal changes in metabolism, which are really quite dramatic. And I have no doubt that there are going to be differences between people who have existed in far northern climes or far southern climes versus someone in Mexico City that's very close to the equator. But we don't have any data on that.
And my second comment was, throughout evolution, the length of the day of the Earth has changed. It was much shorter, like 200 million years ago. It was maybe, 13 hours or something like that, because of angular momentum conservation, because the moon is moving away from the Earth. So I'm wondering if there's anything that can be extracted from that.
That's very interesting. I didn't realize that. Of course, evolution is dynamic and is happening all the time, even today in us, right? And in our offspring and so forth. So I didn't realize that fact to be true, but it's not surprising that we've changed, we've continued to evolve with our environment as the planet and its the speed of it spinning on its axis has changed. That's very interesting. I don't know how to ask hypothesis-driven questions, but it's an interesting observation. Yes, sir.
Do natural short sleep gene risk sleep phenotype in AD mice?
Say that again. Do AD mice?
Those genes that you cross-breed with AD mice, do that change sleep behavior?
Well, they sleep less. The Alzheimer's disease mice on a natural short sleep background sleep less and still do better. That's decidedly different than if you took any of the regular old Alzheimer's disease mouse models and sleep deprive them. Then they get worse much faster. David Holtzman has showed that. We have a lot of work to do in studying the sleep in those. The first pass was to say, is it true, and is it true for multiple genes, that FNSS variants offer genetic protection against accumulation of neuropathology? And now there's just a huge amount of work to continue to do. But an important question. Moses.
It's about glucose metabolism, the link to glucose. I'm wondering with lipid metabolism or protein turnover, is there a link to those events?
Yeah, so this postdoc working in metabolism is very interested in lipids, and lipids as signaling molecules that help to respond to seasonal changes. Because most animals, including us, in the caveman and cavewoman days, would eat as much as we could when there was plenty, because there was going to come a time when there was not an abundance of food. And so we animals are, I think, have evolved to try to capture as much energy. I have a fair amount. I don't want to go there. But having said that, it reminds me to say that you can survive longer, I can survive longer without food than I can without sleep. Right? That's pretty incredible. What are the most important things to us? Oxygen, minutes. Water, days. Sleep, weeks. Food, depending on how corpulent we are, months or longer. So it's pretty interesting to think about how important sleep is and how little we understand about it, about normal sleep, even to this day.
I think we have time for one more question.
Yes, sir.
On the other side of the short sleep syndrome you talk about are people who have mental issues, who have manic depressive. They tend to have short, unstable sleeping. Do you have any thoughts on that?
Yeah, my colleague Andrew Crystal on this slide is a fabulous collaborator, a psychiatrist and a sleep psychiatrist who we've been working with ever since we recruited him to UCSF. And he's one of the world gurus on affect and sleep. And what is clear is that sleep deprivation can trigger an episode of major depression in a bipolar patient. And so bipolar patients who maintain healthy sleep habits do better than ones that don't. I don't know all the literature about what kind of sleep measures have been done in bipolar patients, but it's clear they're clearly linked. Affect worsens with sleep deprivation, and affective disorders contribute to sleep disorders. So again, it's like with Alzheimer's, it's a bidirectional effect. Sleep affecting affect affecting sleep.
Thank you all very much. I apologize, we don't have any more time for questions, but join me in thanking Louis.